Hello all viewers, this is Dr. Neelabh and I am here with the fourth part or fourth lecture in the cancer biology series. In part 3 we have already discussed about proto-oncogenes, what are proto-oncogenes, their definitions and their examples also. We said that proto-oncogenes are the genes which can undergo a gain in functional mutation and lead to cancer. We had discussed about several examples of the proto-oncogenes also. In today's lecture we would continue the discussion about proto-oncogenes. We said that a number of mutations can take place in case of proto-oncogenes. Today we would say that in proto-oncogenes, in some kind of proto-oncogenes, point mutations can take place. What are point mutations? We already know this is the very basic stuff, that point mutations can be of type deletion, insertion, substitution. Now these kind of point mutations generally take place in RAS proto-oncogenes. Such type of point mutations occur and this RAS proto-oncogenes convert into oncogenes and thus cause cancer. So this is what we had to say about point mutations. We just have to remember that point mutations generally occur in case of RAS proto-oncogenes which convert them into oncogenes, thus leading to cancer. Okay. Now coming to another type of change or aberration that occurs in chromosomes. This is the chromosomal translocation. We might be knowing that chromosomal translocation is a process in which a chunk of a chromosome or a part of chromosome is moved from one location to another. So this is the basic stuff. There are a number of methods by which chromosomal translocation takes place. We won't go into the detail of that stuff. We just need to understand that chromosomal translocation occurs. And what is a chromosomal translocation? A part of a chromosome moves from one location to the another. So there is a gene called c-MYC gene. We have already said that in the previous lectures we have said that c-MYC gene is a type of proto-oncogene. Okay, so this c-MYC gene produces, or it is responsible for the production of transcription factors. We all know that transcription factors are involved in the growth and proliferation of the cells. That means that this c-MYC gene is responsible for the proliferation of the cell, that is the cell division, that it promotes the growth of the cell. Okay, so c-MYC gene is responsible for the growth of the cell or transcription or promotion or proliferation of the cell. Now what happens in case of Burkitt's lymphoma? Chromosomal translocation takes place. This c-MYC gene is present on chromosome number 8. This c-MYC gene is present on chromosome number 8. Okay, and after chromosomal translocation, this c-MYC gene is moved to chromosome number 14. I hope that you all might be knowing how to read or how to understand this address. This is the address of a gene. Chromosome 8 means that the gene is present on chromosome number 8. Q here means the longer arm of the chromosome. We all know that the chromosome has two arms, P and Q. P is the shorter one and Q is the longer one depending upon the position of the centromere. Here also chromosome number, this is 14, that means the chromosome number 14 is being talked about here. Q again means the longer arm, and this is the address or the position of the gene that we are talking about. So in case of Burkitt's lymphoma, what happens? This c-MYC gene present on chromosome number 8, Q24 position, is translocated, is moved to chromosome number 14, Q32 position. Okay, so previously the c-MYC gene was being controlled by a different enhancer. This was being controlled by a different enhancer, but now after translocation what will happen is this c-MYC gene moves to a different position and now it is in control of a different enhancer, that is IgG gene enhancer. We all know that IgG gene is continuously expressed. Therefore, its enhancer is responsible for continuous expression of IgG gene. Therefore, when this c-MYC gene comes in control of IgG gene enhancer, the c-MYC gene is also now continuously expressed. That is, constitutive expression of c-MYC gene takes place. Therefore, I have written here continuous expression. Now, if you see here that c-MYC gene is responsible for growth and proliferation of the cell. If continuous expression of c-MYC gene will occur, then what will happen? The cell will go on dividing, the cell will go on proliferating. And what happens when the cell goes on proliferating? Uncontrolled proliferation, it leads to cancer. Okay, so this is the example of Burkitt's lymphoma. We have to remember this can be a good question of part B, that which two chromosomes are involved in chromosomal translocation. In case of Burkitt's lymphoma, it is chromosome number 8 and chromosome number 14. So this is what we have to remember, and also the function of c-MYC gene and what is a c-MYC gene, that is it is a proto-oncogene. Okay, so the next example in this case is chronic myelogenous leukemia. This is also a very important example. In case of chronic myelogenous leukemia, just like the previous example, 9 to 22 translocation takes place. This means that the chromosome number 9, genes present on chromosome number 9 are translocated to chromosome number 22. This is what happens. This is an important example because chromosome number 22 is involved over here. What is chromosome number 22? It is Philadelphia chromosome. We all know that Philadelphia chromosome has been asked a number of times in the previous examinations. So remember that chromosome number 22 is Philadelphia chromosome, and this is involved in chronic myelogenous leukemia. What happens is an ABL gene is present on chromosome number 9. The function of ABL gene is the production of tyrosine kinase. So tyrosine kinase is produced by this ABL gene. We have already studied, or we have already read in the previous lecture, that tyrosine kinase is also the production of tyrosine kinase, or the genes responsible for the production of tyrosine kinase are also proto-oncogenes, because this tyrosine kinase plays an important role in cell proliferation. Okay, so this ABL gene encodes tyrosine kinase, and it is present on chromosome number 9. After translocation, what happens? This ABL gene comes close to BCR gene, which is present on chromosome number 22, that is the Philadelphia chromosome. Please understand this again. ABL gene present on chromosome number 9 after translocation comes near to BCR gene of chromosome number 22, which is the Philadelphia chromosome. The function of BCR gene is not clear. It is unclear till now. But what is seen here, that the ABL gene alone produces a limited amount of tyrosine kinase that can be used, or that is useful for the cell. But after ABL comes near to BCL, a BCR ABL fusion gene is formed. What happens is, after this fusion gene is formed, it is involved in continuous expression of tyrosine kinase. A lot of tyrosine kinase is produced, a continuous expression of tyrosine kinase is produced, and we have already read that tyrosine kinase can be an important proto-oncogene, or the genes responsible for the production of tyrosine kinase can be proto-oncogenes. So this continuous expression of tyrosine kinase, it will aid in the regular proliferation or a lot of proliferation of the cell, and ultimately cause cancer. So this is the ultimate product, or the cancer would be caused after the regular expression of tyrosine kinase. So this chronic myelogenous leukemia is another example or chromosomal aberration that happens in chromosome 9, where the gene ABL is present, and it moves to chromosome number 22, which is Philadelphia chromosome. A fusion gene is formed, which is again involved in the continuous expression of tyrosine kinase. Finally, we would move to insertional activation. This is we were studying chromosomal translocation up till now. We studied Burkitt's lymphoma, the first example in this case, and chronic myelogenous leukemia, the second example in this case. Now we are coming to insertional activation. This is altogether a different kind of stuff. In case of insertional activation also, c-Myc protogene is an example. c-Myc proto-oncogene has 3 exons, E1, E2 and E3. These are the 3 exons found on c-Myc proto-oncogene. Okay, we have already studied that c-Myc produces transcriptional factors, that is, it is responsible for the cell proliferation. Okay, now there is an virus called as avian leukosis virus. In short, it is said as ALV. What happens is ALV, this avian leukosis virus, integrates its DNA, integrates its stuff between the exon 1 and exon 2. Now I have shown this in red color. You can see that ALV integration is shown between exon 1 and exon 2. Now what happens, the promoter of this ALV takes charge of E2 and E3 exons. What happens is the promoter present in the ALV takes charge of E2 and E3 exons, and it increases the transcription of E2 and E3. This ALV promoter increases the transcription of E2 and E3, which again will definitely increase the synthesis of c-Myc protein. We have already said that c-Myc produces transcription factors. That means that growth and proliferation, a lot of growth and proliferation will take place after this occurrence, and again it will lead to cancer. So this is another kind of stuff in which ALV, that is avian leukovirus, integrates its DNA between exon 1 and exon 2. This ALV promoter takes charge of exon 2 and exon 3 and increases its transcription, and which in turn increases the production of c-Myc, which in turn again causes cancer. So these were the examples, or these were the mechanisms by which mutations occur in case of or changes or aberration occurs again, which cause cancer in proto-oncogenes. So this was all about this proto-oncogenes. In the next class we would take up tumor suppressor genes. I hope you understood this lecture quite well. If you like this video, press the like button and subscribe to my channel. Thank you and have a great day.